Hi learners, welcome back. So on this slide, we continue with EKC with complex equilibrium under the CE here. I would like you to focus on these two parts here. Alright, so again, this is inside the capillary of the CE system here. You have your cathode and anode, and inside the capillary, you have the cations first, is your analyte. But due to the presence of ligand, they will complex, to form this, a complexation. Now, of course, with the binding of a ligand and a cation, we have this binding affinity. And of course, we have a kind of equilibrium constant, capital K. So we can quite Ka association constant. It's given by B over A, which A is denoted by the molar fraction of ions in the free solutions, and B is the molar fraction of ions associated with the ligand. And the reason is because we begin first with cations, and then you add the ligands, right? So we begin, the starting state is the cation, and therefore is always at the bottom, the denominator. Now, in terms of mu observed, it is just a sum of the molar fraction of the ions multiplied by the own mu, and B as a product of this mu observed of the ligands themselves as a free ligand. Okay. So you can see that here, analytes that's not complex with ligand, they won't be held back because it's just their electroosmotic flow and the charge interaction, it just move forward. But on interactions with the ligand here, it will be pulled back by the mu EP. Here, we introduce this term called SELEX. You hear me talking about SELEX, CE-SELEX, non-SELEX. And SELEX is really just an acronym for systematic evolutions of ligands by exponential enrichment. SELEX, S-E-L-E-X. What is it? So pretty much it's a kind of a pre-concentration and kind of enrichment, because we have a mixture of all your analyte, and you're trying to use your target to just absorb those that can bind and removing the rest here. So it uses this partition system where it has a binding affinity to a certain specific target, of your mixture in the library, and then removing the rest. And when you go through the system again and again, you can really enrich what you want to scan. So in a typical SELEX procedure, you have a nucleic acid kind of library, to a target, and this target normally we call like a CE-SELEX here inside a capillary, so capillary electrophoresis. And then we separate all these particles here by the binding, unbinding is a detachment of nucleic acid. So those acids that are not bound, will be removed, and therefore through this process we can amplify the amount of these nucleic acids for the PCR reactions here. Okay, and we do it many times, the whole amount will be enriched. Now, there's certain advantages and disadvantages using these aptamers compared to the antibodies here. So one thing to note is, as we discussed earlier in this module here, using of animals might not be ethical. It's a bit harder to clear your ethics approval board here. So for antibodies, unfortunately, we need the use of animals for testing. But for aptamers, it's not. It's more of lab-based, and we do it in the in vitro process. Okay. Next thing is, the labeling of antibodies could cause losses in affinity, But in aptamers, because it's produced, so it can be attached to the aptamers at precise location, not involved in binding. Now let me go back to the top here. Let's focus on more of the chemistry side of the house. For antibodies, there are limitations against targeting certain parts of the body or the toxic substance. But aptamers, how they function is, some toxins, we can use them, and they do not generate good immunoresponses from antibodies, but inside aptamers they have very high affinity. So you can, in general, think of it like aptamers are more of a man-made, we manufacture it and we engineer it to the target as we want, but antibodies are a bit more natural in that area. I'm just using an analogy. Now the other part is, for antibodies, they have this certain restriction here. So the kinetic parameters of this AB and AG, they can't be changed as we want. But for aptamers, again, because we control it, we can turn it off and off. So Ka, the rate constant for the association, and Kd, the dissociation constant, for the rate. Now antibodies also have a limited shelf life, because again, you can think of them like a life, life. and they're very sensitive. They can be denatured at high extreme temperature, right? But for aptamers, right, even if they're denatured, they can come back again. They can respawn or require regeneration, okay, through minutes, because they are just bondings, and so you can harness this durability, having a long-term storage, which means you don't have to purchase again and buy. You can keep for a long time, and you can, moreover, be transported from one lab to another lab, even overseas, at ambient temperature. Antibodies is much harder. Next is now the identification of antibodies, right, to recognize targets, right, under certain conditions, not in the physiological conditions, is not feasible. So antibodies again is more about life. Think of like a life like human, right? That is, we function at 37 degrees Celsius, so it's very specific range. We have a high temperature, we get fever, we don't function anymore here, it's not good. But again, aptamers is like no life, it is a man-made. So you think of it like a robot, right? Robot, you can just tune it, right, to a temperature, to certain parameters that's not even within the working range of antibodies. Now, antibodies again, because it's more human-like, right? It's more alive, so it might have some variations, from different batches. But aptamers, because they are controlled and produced in chemical synthesis, right? So there's a highly reproducible in every batch. Okay, here are some true examples. Here I'd like you to compare when in the first diagram here, right? So this is an electrophoric diagram, right? Where here the difference between the first and second line is just that, right? The first is just a pure DNA library, and after that you have this addition of the MRCK, which is a form of this enzyme, right? And then you see that this is the window where you can collect your aptamer. Okay, you see some binding affinity, right? Then you see some signals there. Now the next one here, right, this using another concentration of the active MRCK, right? Here's really enriched aptamer. So here you see that there's certain signals derived from the complexations. And the last one here, you see that there's inactive and also active, this kinase, a form of enzyme. So you see that even if we add the aptamer, but if it's inactive, right, because the site is being, blocked, right? Again, there's no complexation and you do not see the peak here. So a shorthand you might want to know is called the NESIM, is the non-equilibrium capillary electrophoresis of equilibrium mixtures. Okay, again, this is about partition phase chemistry, and we try to use the free aptamer and the complexations. Okay, here another diagram here, some schematic sample. So you have different parts, right, in this equilibrium mixture, right, and then at time equal to 0, right, you inject this injection mixture, some of the L, C, and T here, and then slowly they migrate in different speed, and then now after that you see a distribution because they dissociate, right, and then you see that the high concentration will be at the end because they move faster. This is how we separate them using the NESIM. Some legend, the L is the free ligand, T is the target, free target, and then C is the complex, okay, combined together here, because we know that in the first slide we have associations, dissociations. Okay, this is another one here. We look at how an example of this NESIM, right? The interactions between a fluorescent labeled DNA ligand, and in class we also talk about how we use the fluorophore, right, attachment to your substrate here. So in this case, we are tagging this fluorescent dye to a single stranded binding protein, right? And of course, because there's some kind of fluorophore involved here, there could be some decay, right, because of the complexation. So we want you to see that, right? So there's this equilibrium reaction occurring between the complex formation and dissociation. So it's sort of like a stretch, pull and push. Okay. And this is more of the kinetics part. Some form of equations. You see this again, the dissociations constant, a kind of equilibrium constant, and you can just substitute the concentrations of the various particles there. Of course, you can have it. So what you would need is really the initial concentration and the peak area to do these calculations here. I know this is a bit more life science, but I think it's not that hard. ATGC, right? Your basis. Okay. I want you to focus there, right? For low dissociation constant, it means stronger binding, because after you have a high dissociation constant, it doesn't want to dissociate, so it binds very strongly. That's why it has a low dissociation constant. Okay. Next part here, I want to introduce something called the MAB. MAB, okay, MAP is the molecular aptamer beacon. So what it does is, right, so at first you see, before that, right, this particle here doesn't fluoresce, because the fluorophore that's supposed to fluoresce is closely attached to the quencher. Quencher means that it just want to fluoresce but just doesn't allow it. But in the presence of a protein, because of the various binding, the bases are separated, then now the fluorophore stands alone. It can function, right, and beam, right, and fluoresce, because now the quencher is very far away. So that's how it works here, and we call this the hairpin structure. Okay, you try to detach this bond here, and remember these bonds are all of hydrogen bonding. Okay, so it's using this bonding of chemistry to do something very advanced. Another example, because they are just separated by this hydrogen bond here, you can also have a attachment, right, of another aptamer, right, together. Okay, so it just it looks like a diamond to you, but I want you to see that it is interacting at the key, the portion. Okay, here you have this graph, which in the y-axis is the RFU, it's just relative fluorescence unit, because we are using a laser-induced fluorescence detector to measure how much it fluoresces. So we see a tapering. This is asymptote, because once you reach saturation, there's no more increased fluorescent emission. Okay, of this MRCK MAB. Okay, MAB, remember, is a molecular aptamer beacon. Okay, so we know that this is the part that has reached saturation. Okay, and then I'll continue in the next part. Cheers.